The next biochemistry exam question we'll be looking at is students were asked to write a note on proteins that function as enzymes. Now, let me tell you something. If they ask you to write about proteins that function as enzymes, this is what they want to see. And what is the mnemonic for understanding these types of enzymes that are actually proteins? The mnemonic is what? Otleo, 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 like this. O is for oxidoreductase, T is for transferase, H is for hydrolase, L is for lyase, I is for isomerase, L is for ligase. So don't worry, we'll come to this, right? So let's start from this process. In fact, if you talk about this thing and those examples are not there, forget about the marks. Okay? So we say that proteins that function as enzymes are called are called what? Enzymatic proteins. Okay, that was a typo. Proteins that function as enzymes are called enzymatic proteins. This must be your opening sentence. Okay, so what are their characteristics? Okay, so they have what? Catalytic activity. They accelerate chemical reactions. They are enzymes. They help chemical reactions to actually what? Progress faster. Substrate specificity. That means what? They don't just act. They are specific. Okay, the oxidoreductase will basically what? Enhance oxidation reduction reactions. Transferases, these ones will transfer a group. Do you understand? Those ones will transfer a group. Hydrolase, that's one they will basically add water. Lyase, they will break down. Do you understand? They are specific. So they bind to specific substrate. They have high efficiency. That means they have what? Rapid reaction rates. Okay. Reversibility. They can catalyze reversible reactions. Okay. So these are the characteristics. Just grab them. The mnemonics to understand these characteristics is what? CSHR. Those first letters there. CSHR. I can see clearly remember it. Okay. So now talking about the types now. Oxidoreductase. Examples of oxidoreductase is what? Lactase dehydrogenase. You don't have to start grabbing like two. Just grab one. Okay. Oxidoreductase is one. They catalyze oxidation reduction reactions. Lactate dehydrogenase is an example. Transferase. This is an oxidoreductase. This one they catalyze what? Electron transfers reaction. That's either oxidation or reduction. Okay. So you can see how the electrons are basically what? Transferred. Electron is now transferred from this molecule to this molecule. Okay, there's a transfer of electrons. Then transferases is what? Like one example is what? Glycogen synthase. What is a transferase? Transferase would catalyze the transfer of a functional group to another molecule. All right, that other one was electrons. This one is functional group. Okay, example is glycogen synthase. Then we have the hydrolase enzymes. Okay, one of example is what? Lactase. What is hydrolase? This one will break the bond by hydrolase. If this thing is joined together, okay, if you add water, it should break it down. Like for example, if you add water to salt, the salt will become liquid, right? Shows that there is an hydraulic reaction that just went on. Then lyase. An example of lyase is what? Aldolase. Lyase is what? What is the use of lyase? Lyase. I didn't bring a picture for lyase. But lyase is basically what? Enzymes that break down to. But this one they don't use water. Then isomerase. An example of isomerase is what? Phosphoglucose isomerase. Alright. So isomerase is basically enzymes that bring together isomers. You are bringing two isomers together to join them. Alright. The ligase. Ligase is like a breakdown, but this breakdown is actually what? Specific to DNA. Alright. So ligases are actually what? Molecular glues. Sorry. They bind molecules together. Sorry, the ligase they bind. They bind together. Okay, they actually bind together. All right, so binding two strands of DNA together is done by a ligase enzyme. All right, so examples of ligase enzyme include what? DNA ligase. right? So other examples of enzymes that are protein enzymes, I told you that there are many examples, but you want to see this, right? Other examples include digestive enzymes like amylase for carbohydrate digestion, protease for protein digestion, ligase for lipid digestion. Then okay, that's under digestive enzymes. Then for energy metabolism enzymes, we have the pyruvate kinase, citrate synthase, ATP synthase, right? Biosynthesis, what? Biosynthetic enzymes, we have the DNA polymerase, RNA polymerase. So if looking at this question, the important thing was these characteristics, then this thing. This one is optional, all right? Then what is the structure of a protein enzyme? They have an active site. You must actually talk about it. It's a substrate binding site, okay? Have an active site, okay? Then also have a binding site, which is a region for cofactor or regulatory molecules, all right? So if there's a molecule that is supposed to come in to regulate the process, can bind at the binding site. Then catalytic domain, this is a region that is responsible for the catalysis, all right? So that's just a note on proteins that function as enzymes.